So today's speaker is Dr. Karine Roch. She's a professor of molecular systems biology, and she is going to talk to us about a systems biology approach to understand gene regulation, pathogenicity, and human malaria parasites.
So we just had a retreat, so I presented the data that I generated for the mosquitoes, so I thought it would be good for you to also know what I'm doing outside mosquitoes and what I am doing on the human malaria parasites. So a short introduction. I'm pretty sure you all know malaria is bad. It's a mosquitoes-borne disease, so we still have the mosquitoes here. A common interest. One of the deadliest infectious diseases in the world. 198 million cases per year. And the number of deaths have decreased significantly over the past 10 years. We went from millions to 430,000 deaths annually, but it's still one of the main cause of death in sub-Saharan Africa. There's no effective FDA-approved vaccine, and it's really not protective. It's actually just protecting against the most hard side effect of the disease, and the biggest issue is the resistance of all anti-malaria, including artemisinin. So for those that are not really aware of the malaria parasite life cycle, when an infected mosquito bites a human, he injects what we call sporozoites. The sporozoites goes quickly into the liver and then differentiates and replicate intensively, and within 10 days, the parasite leaves the liver and infects red blood cells. In 48 hours, the parasites transform from a merozoite to a ring stage, to pozoite and schisant, and within 48 hours is going to give 16 to 32 new parasites. This is an intensive stage of replication. This is also the symptomatic phase of the disease. At some point when the parasite sends some stress, this intensive replication process stops and we have a sexual differentiation into mature male and female gametocytes. And when a mosquito bite, you inject this sexually mature gametocyte into the mosquito's midgut, where there will be sexual differentiation. And then again, sexual reproduction and differentiation into different stages. And then the new sporozoite will travel to the salivary glands and infect a new element. So my lab has been really working on the whole life cycle of the human malaria parasite. We're trying to understand how the parasite regulates all these different stages at the transcriptional and post-transcriptional level. And we mostly worked in the asexual and sexual stage, which is easiest to work with because you can actually grow the parasite in culture, but we're also trying to understand what's going on in the sporozoite and liver stage also. And the idea will be really to block asexual multiplication within the red blood cell stage, sexual reproduction here, to block transmission of the disease from human to mosquitoes and eventually mosquitoes to human as well. So the malaria parasite genome has been sequenced more than 15 years ago now. It's similar to the yeast, I would say. There is 23 megabase in size, 14 chromosomes. There's also one mitochondria and one plastid. So Plasmodium is similar to a plant-like organism. There is approximately 6,200 genes, from which 5,500 are coding genes. It's still one of the most 80 eukaryotic genome sequenced to date. And it's the reason why it's sometimes extremely difficult to work at the molecular level. So for the past 15 plus years, I've been now working on generating a lot of genome-wide data together with other collaborators in lab. So we now know when a transcript is expressed across the whole parasite life cycle. We also know when a protein is present. People have been working on trying to understand protein-protein interaction and also starting to grasp a bit better the metabolome of the malaria parasite. However, we know when the gene is there. We know that the transcriptional profile is tightly regulated across the whole parasite life cycle. However, gene regulation in the parasite is really not well understood. while we know and all the general transcription factors are present in the malaria parasite genome, the specific transcription factor, which are known to be extremely important to specifically transcribe gene when needed, there is a lack of transcription factor in the parasite genome. Only 27 specific transcription factors of the AP82 family, which are plant-related, have been identified and validated as transcription factor in the parasite genome. So it's still a very low number when you compare to human or Saccharomyces cerevisiae. We really miss 1/3 of the transcription factor needed to regulate this cascade of transcripts. So my lab over the past 15 years, I've been really trying to understand gene regulation at the chromatin structure level, but also post-transcriptional level. So if we want to understand gene regulation in classical eukaryotes, we know that transcription factor and DNA binding protein are extremely important at the local chromatin structure level. We also know that nucleosome positioning are important to compact or unfold chromatin and allow the transcription factor to interact and transcribe gene when needed. So this has been pretty basic over the past, I would say, 15 years. And recently, people have focused a bit more on trying to understand the global genome organization. So how the chromosome and genome are organized at the 3D level, and how this organization into domain-like structure or chromosome-chromosome interaction can actually influence gene regulation. So this is global genome organization that involves nuclear compartmentation, chromatin looping, and as well as long-range DNA interaction. And my lab also worked at the post-transcriptional level, but I'm not going to spend too much time on this because I don't have enough time. So I'm really going to concentrate on the work that my lab has been done on trying to understand change in nucleosome positioning, histone modification, as well as a structure of the chromatin to understand a parasite development within the human or within the mosquitoes. So the first thing that we started was already many years ago, where we tried to understand changing nucleosome landscape across the parasite life cycle. We use a very simple methodology. I'm not going too much into detail, but we actually try to purify nucleosome-free and nucleosome-bound region. And all of these DNA, purified DNA, nucleosome-free or nucleosome-bound, were then sequenced using the first Illumina sequencing machine and mapped into the genome. And then after that, we tried to correlate change in nucleosome positioning across the different stages of the life cycle progression with mRNA steady state level. And this is a correlation that we've tried to draw between nucleosome landscape and mRNA steady state level. And you will see that the correlation is not perfect. We have some gene that seems to be closed across the whole life cycle progression, and some gene that seems to be always open. But in general, we have a binary structure of the chromatin where the chromatin is condensed and then open up and then condense again. And there is a lack of correlation between nucleosome positioning and mRNA steady state. What we found, however, is the amount of nucleosome in the promoter region. So here is the transcription start site for each of the gene average. So in peak, you have a very high level of nucleosome positioning in the promoter region of each gene. So when we have a very high level of nucleosome, we have a very low level of expression. Okay, this is the expression level, so very low. If we have a very low level of nucleosome positioning, then we have a high level of expression. So nucleosome positioning is important to regulate transcript level, but change of nucleosome positioning doesn't seem to be that well correlated, at least for all the gene with change in gene expression profile. So chromatin organization and promoter region correlate well with gene expression activity in general. So we came up with this picture where we think there's a huge depletion of chromatin structure happening at this particular stage, trophozoite, when before the parasite is able to or start its intensive replication. But at the beginning, you actually see here, that's the red blood cell, that's the parasite. There's a very intense, dark purple color here, which corresponds to the nucleosome, and intense coloring of histone H3. So the chromosome is heavily compacted. There is a lot of nucleosome here. Then when the cell cycle starts, the parasites open up, start this intensive transcriptional activity, and you actually see the nucleus opening up. There is a decrease of histone level, however, there is an increased transcriptional activity. This is a IFA response 2. And then when the parasite in the schizone stage, you start to see a more nucleosome positioning and a decrease in RNA pol 2 activity. So we have a very nice correlation of all of these data sets that show that when the chromosome is compacted, there is not much going on. When the chromosome open up, there is a lot of transcriptional activity, and then a decrease of transcriptional activity. So we think that nucleosome position is extremely important to regulate active transcriptional regulation here, and the cascade of transcript that we see is a combination of transcriptional activity and transcriptional stability of all of the transcripts that have been mostly transcribed at this particular stage. So to validate this model, we actually adapt this chromosome conformation capture to the malaria parasite. So it's called IC, where we cross-link directly the DNA and all the protein at different stages of the life cycle. Within the nucleus, we then digest the chromatin with the restriction enzyme, and then ligate within the nucleus again the DNA fragment that will be found in close proximity. So it's in fact a very simple type of technology that will allow every single DNA fragment that are supposedly far away within the linear chromatin to bind together. They are in close proximity into the 3D structure. So we do paired L reading on that, and all with pair are then mapped to the genome and merged into what we call contact conch matrix. So that may be a bit difficult for those that are not familiar with these matrices. This is an intra-chromosomal contact conch matrix, so I think this is chromosome 7 of the malaria parasites. This is one end of the chromosome, this is the centromere, the other end of the chromosome. And basically every single DNA fragment that interact with each other next to each other has a very bigger chance to interact together. So you have a very high level of contact counts and ligation that happen at that level. And if you're far away at the 3D level, then there is not much interaction between, I would say, this region here and this region here. However, what we notice in this particular case is we have the telomere end that interacts strongly with each other here because you have an increased correlation between both of these telomere ends. And I'm going to talk about that a bit later. This genomic region here interact also strongly with the chromosome ends. So this is one single chromosome and contact conch matrix for intrachromosomal contact. And this is actually the interchromosomal contact count. And you will see that each chromosome has, of course, a much more high probability to interact with each other than with another chromosome. But you can see here, this region heavily interact with chromosome 1, and it happened to be actually the centromere of chromosome 13, for example, with chromosome 1, increased correlation. So if you get familiar with these matrices, you usually stare them a lot and try to understand what's going on, and after a while, you start to see biology opening up. So I like to compare IC technology to the fluorescence microscopy. When you do DNA fish experiments, you try to localize and validate two different probes against each other. And this IC technology really allows you to map millions of probes against millions of probes. And instead of having just a localization of two different genes or two different locus, you can actually model at the genome-wide level the 3D structure of your genome. So we have done that type of experiments now for the human malaria parasite Plasmodium falciparum, but we have also tried to generate additional IC data across the whole malaria spectrum, so Plasmodium infecting monkey, Knowlesii, or different Plasmodium species infecting a mouse, the mouse model. And we also tried to compare this genome structure to Toxoplasma gondia, which is a related apicomplex and parasite, also Babesia macroti or Babesia doceni. to see if there is any particular feature that this apicomplexan parasite can share, and if we can actually highlight a general feature of gene regulation and also a general feature of pathogenicity. So we've been doing all of these experiments, and this is just an example of what we've done for the Plasmodium falciparum. So again, cross-link, biatylated the protein, digestion by restriction enzyme, ligation, purification, and then live preparation and generation of all of this map. And then after that, we stare at the map. So this is chromosome 2. Again, chromosome 2 against chromosome 2. One end of the telomere, the centromere of this particular chromosome, and the other end of the telomere. And you can see this classical X shape. And then when you do the 2D model or 3D model, basically, you see that the centromere is at one end, the telomere at another end, and the telomere tend to interact with each other. So we've seen this feature that really characteristic of what we see in budding and fishing yeast, this folded chromosome architecture encore at the centromere. And this happened for most of the chromosome for all Plasmodium, Toxoplasma, and Babesia species, I would say. However, for Plasmodium falciparum, we observe some very atypical feature that's not observed in other apicomplexan parasites, this domain-like structure. And this domain-like structure happen to be present for all gene involved in non-genic variation. So Plasmodium falciparum has gene involved in non-genic variation that are extremely important for the parasite to avoid host immune system. They are usually localized, all of these gene family are localized at the telomere ends, and some of these gene family are actually within the chromosome, so chromosome 4, 6, 7, 8, and 12. And for all of this one, we see this really classical domain-like structure that show that you have an additional domain, a looping, to make sure all of these gene family interact with each other and certainly communicate with each other. So just to give you a bit more explanation, this non-genic variation encoded by 60 different genes, only one is expressed and exported as a surface of the white blood cell to avoid the host immune system. So when the host recognizes this VAR gene, the parasite is going to switch and express another one. And this is very important for the parasite to avoid the host immune system and mediate adherence also of the infected red blood cell to the endothelium. So understanding how these genes are regulated at the transcriptional level is extremely important for vaccine discovery of new or development of new targeted therapy. And again, it's really classical. You really see that all of this VAR gene and all of these genes involved in pathogenicity and gene variation really interact with each other in all of these matrix. And this interaction is conserved across the different stage of the life cycle. This is the ring stage, trophozoite schizons. It's also present in the mosquito stage and also present in the gametocyte stage. And if you zoom in, you actually see additional feature coming up at different stage of the parasite life cycle. So we were very happy about that. And then the other feature that we actually see is if we compare all the other malaria parasite species that we have done so far, we only see this domain-like structure in Plasmodium falciparum and Plasmodium knowlesii. And these are the only two Plasmodium species that have this non-genic variation family. So this VAR gene in Plasmodium falciparum and C-cabir in Plasmodium knowlesii, and are the gene family that are responsible for death in human patients. So you don't see any of this structure in Plasmodium myolii or Plasmodium berghii. You only see this classical X-shaped structure that is also observed in yeast. But this really seems to be characteristic of the most pathogenic strain of malaria parasites. So we're really excited about that. We also validate that this telomere end and also this domain-like structure correlates at the 2D level with certain histone mark. So they are marked by a repressive mark. So basically histone H3 K9 methylation. This is a chromatin IP experiment where we try to localize histone repressive mark across the whole parasite genome. So here's a zoom in of the telomere ends. Here you have all the virgin, and that are marked here, chromosome 2, all the virgin are marked, interact with each other, and basically this is a heterochromatin mark, so that repress all of this gene family. We also validate that using IFA experiments. Another very interesting feature that we observe across the different stages of the life cycle, this is the chromosome 2 for trophozoite stage, so the asexual stage. And this is chromosome 2 for the gametocyte stage. And you will see that this is the telomere ends interact nicely here, but during sexual differentiation, we have an increased interaction here, and increased amount of heterochromatin, strong close chromatin structure. And if we validate using ChIP-seq, it's on H3 canine methylation. This is the asexual stage. You see very nice histone methylation mark here. And during the sexual stage, we have an increased expansion of this heterochromatin structure here that corresponds to an increased compaction at the 3D structure level. And these are histone marks that are marking the virgin. And here, actually, very interesting, it's genes that are expressed and exported at the surface of the red blood cell. And these genes basically are not needed in the mosquitoes anymore. Parasites don't need to infect red blood cells. These proteins don't need to be exported at the surface of the red blood cell, so they are actually switched off and come and interact with a repressive center here. Another very interesting feature, again, when the parasites invade the red blood cell, there is a large number of invasion genes that are expressed that need to be intensively expressed to help the parasite to invade the host red blood cell. And it's on basically during the asexual stage, during gametocytes or sporozoites, so within the mosquito, these genes are actually coming and interact with the virgin and the repressive center. So basically it's active during the asexual stage, and they come and interact strongly with the repressive center during the sexual stage or the mosquito stage, because again, these genes are not needed anymore in the mosquito stage. And this is the opposite example. So this is chromosome 12, I believe. So this internal virgin cluster that you see, they all interact together. And so this one internal cluster, another one, and here there is a dot here. This is a gene that is very important. It's a transcription factor that is activated during sexual differentiation. So basically this gene here interacts strongly with the telomere end, with the repressed gene virgin, but then during sexual differentiation need to moved away from this inactive center into the active stage, and you lost this dot and this repressive here during sexual differentiation, which again makes sense because these genes need to be turned on during sexual differentiation. Another very interesting feature that we also find last a couple of years ago now, this is chromosome 14. During the asexual stage, there is nothing really exciting. There is just an X shape. There is no internal virgin. No gene involved too much in pathogenicity is in this chromosome. However, during gametocyte stage and also in the sporozoite stage, I didn't show the picture here, in the mosquitoes, we have a very intense compaction here with this large superdomain that we see, specifically in the gametocyte stage. And we test different type of gametocyte stage, and we always see this structure. And if we zoom in, it's clear that there is a lot of compaction, domain-like structure that happened during the sexual differentiation. And this for us was a reminiscence of this super compaction that we see during X inactivation of mouse or human X chromosome. We have two X chromosomes if we are mammalian and female, and one of the X needs to be inactivated for the dosage compensation. And this is really something that we observe also in the malaria parasite during sexual differentiation. So we tried to zoom in and see what was going on at the boundary of these two superdomains, and we find a transcription factor that's possibly involved in a gene regulation in the mosquito stage. So we actually decided to tag this specific transcription factor in the genome, the GFP, and follow the expression of the gene across sexual differentiation. And this is a sexual differentiation. We have two male here and a female here. And basically, we only see this transcription factor expressed in the female mosquito gametocytes, not in the male. And we really think that the formation of this change in chromatin structure may be important to regulate male and female differentiation across the parasite life cycle. So this is something that we're following up to understand exactly what's going on here. So the next step that we really wanted to understand is we find some very nice correlation between gene involved in pathogenicity. They correlate very well with each other. But then we wanted to see a bit more if change in gene expression really correlates with change in chromatin structure that we see at the matrix level. So we then model all of these contact conch matrix into a 3D structure using a mathematical model. And then we came up with different 3D structure for the wing, the trophozoite and schizoid. And here again, we actually see and model a very strong compaction, opening up of the chromatin structure and compaction again. And we observe, I think I have here, that the centromere all correlate with each other. That's something we saw already into the contact conch matrix. All the telomere, virulent gene, or repressed gene seems to correlate also with each other. But then we move on, so there's a very strong correlation about this repressed gene. But then, so we also validate this 2D structure using DNA fish to make sure our 3D model was actually accurate. So using two different colors for two genes close to each other, and we validate this type of model for a different set of genes. And we also did similar type of approach for all of the different Plasmodium strains that we looked at across the different stages. So Plasmodium falciparum with trophozoite, gametocytes, sporozoite, we also have Plasmodium vivax, Toxoplasma, Gondii, Babesia. And in general, we really observe the conservation of this 3D structure. Centromere tend to cluster together. Telomere and virulent gene tend to cluster together for a lot of species, with the exception of Toxoplasma gondii, and it happened that we can survive extremely well with Toxoplasma. There is no gene variation, and there is no clear correlation between pathogenicity and gene structure. We still see this nice structure in Babesia, so we think there is some virulent gene there. For Plasmodium knowlesia, the 3D structure model was not really nice, but you still see that the centromere cluster together. There is so many virulent genes for this particular Plasmodium that it's really difficult to actually show you in a 1D, the 3D model, but there's still a nice correlation with genome organization here. And then when we try to superpose 3D structure and gene expression profile, this is data show for Plasmodium falciparum. So we basically took every single gene expression profile that people have generated in the field, average using a vector matrix, all the gene expression profile, and then superimpose the gene expression profile of each gene on the 3D structure. And you clearly see large repressive center here, and then if you move away from the telomere end, you actually increase in general transcription levels. And this is also something we did for all the different Plasmodium species, and we were happy to see that it's really conserved. This is Plasmodium falciparum, this is the telomere end, and if you moved away from the telomere end, you see an increased transcriptional regulation here. This is also true for Plasmodium knowlesii. Picture here is not really beautiful, but you have a repression, and then at the opposite, very intense transcription there. It's not true for malaria parasites infecting mouse. We really didn't see any strong correlation there, but it's true for Babesia, and we have an opposite picture for Toxoplasma gondii. Basically, you have a high expression at the telomere end, and you have a repression at the centromere end. So very opposite picture for this parasite here. But overall, there is a very nice correlation between our 3D level and the gene expression profile. So I would like to conclude this first part The change in chromatin structure is particularly interesting for parasite-specific gene involving pathogenicity, all cell invasion, and master regulator of transcription. That's what we've seen clearly in all the examples I gave you with Plasmodium falciparum. There's a really nice spatial genome organization in most Plasmodium species, and it's really constrained by the localization of virulence gene for Plasmodium, mostly or Babesia, but this is not something we see in Toxoplasma. Distinct change in genome organization during the parasite life cycle correlate with gene expression. And the very interesting additional feature is the two most pathogenic Plasmodium parasites, falciparum and Riverside, are really strongly constrained by their additional gene family that are involved in genetic variation. And if we understand how this is regulated, we may be able to define new vaccine strategy. And basically, our results really indicate a link between genome organization and gene expression in most virulent pathogens. So next step is great. We've seen a very nice correlation between chromatin structure, chromatin domain-like, and gene regulation. What are the molecular components that can be parasite-specific that we can target for novel therapeutic intervention? And basically, we know that proteins are extremely important to regulate this feature, but long non-coding RNA also are important to regulate gene regulation. So can these components be identified in a genome-wide manner and targeted for anti-malarial intervention? That's what my lab is now working on. And we're really trying to focus and to follow what people are doing in the X inactivation chromosome field, where they try to identify these long non-coding RNA, such as the X inactivation long non-coding RNA, or these proteins that are extremely important to really condense this chromatin and this chromosome during cell differentiation. So we started by looking at long non-coding RNA in the malaria parasites. We know that in other eukaryotes, they're extremely important to regulate transcriptional regulation, to enhancer, to chromatin, to also to bring a chromatin modifying enzyme. So extremely important in terms of transcriptional regulation, and additional presence of long non-coding RNA in Plasmodium could actually compensate for the lack of transcription factor. There's also a lot of different function of this long non-coding RNA into the cytoplasm, but I'm not going too much into this right now. Are these long non-coding RNA important in apicomplex and parasites? So this is what we wanted to figure out. So we've been searching intensively a way to identify the presence of potential long non-coding RNA. So some previous labs have done a deep sequencing to analyze potential long non-coding RNA, but we wanted to develop different type of approach. And we first looked at our change in the histone modification. So I talk about histone H3K9 methylation that's important during different stage differentiation, with an extension of this chromatin mark to a press gene involved in invasion or gene exported at the surface of the red blood cell. And during this process, we actually identified some change in histone modification that goes in the opposite direction. So in chromosome 9 or chromosome 14, for example, we actually saw a decrease in histone modification. And this decrease in histone modification did not actually correlate with a change in with a specific gene family here. We have a large histone mark here during asexual stage. This histone mark disappeared during sexual development, but there's absolutely no gene under that. It was similar to this chromosome 14, so we tried to validate that and zoom in into this region. This is the histone mark during asexual sexual stage. We see RNA-seq, and we clearly see an increased level of RNA-seq. At this particular stage, we validate using a quantitative PCR for both of these features that do not correlate with any specific gene here. This is an inter-gene equation. So we think we have at least two candidates to regulate cell differentiation at two long non-coding RNA. But I like to do genome-wide type of data. So we really started a very complex process to decipher the non-coding code of pathogenicity and sexual differentiation in the human malaria parasites. And we developed a very strong and novel computational approach to self-fractionate and separate every different nucleus and cytoplasm, do mRNA isolation in different compartments, and also use growth seq, so RNA that have been specifically purified from nascent RNA that tend to also show transcriptional activity and seems to identify different type of RNA as compared to the RNA-seq here. So we've been doing this intensive computational and transcriptional data analysis to identify long non-coding RNA, and we identified approximately 1,094 long non-coding RNA in the Plasmodium genome. 36 have been previously identified with 64% completely novel. So when you have novel things, you looked at the change in gene expression profile, and similar to what we have in higher eukaryote, we have a clearly change in gene expression across the different stage, either in the nucleus or in the cytoplasmic fraction. And these tend to tell us that there is definitely a lot of regulation going on here, and certainly a lot of function. So we went on and tried to validate using RNA fish the localization and the gene expression profile of these different long non-coding RNA. So this is a long non-coding RNA that's similar to the TAR, which is a long non-coding RNA that's known to be important to cluster telomere and to keep the telomere end happy in higher eukaryotes. So we've been able to localize that definitely into the nucleus at different stages of the life cycle. It's a long non-coding RNA that we call 13 that we detect present in the ring and trophozoite stage, but in our gene expression data was not present in the skeleton stage, and we indeed show that there is no presence of this RNA in the skeleton stage, and different type of other ones that are not present at the ring but present at the troph or skeleton stage. So we validate a lot of that using basic RNA fish experiments, which is great, but doesn't tell you the function. So we developed novel type of approach, genome-wide approach to understand the function of this long non-coding RNA, the malaria parasite, which is called chromatin isolation by RNA purification, CHOP-seq, where you actually, once you know the sequence of this long non-coding RNA or the presence of this long non-coding RNA, you design batch-related oligo that interact with the long non-coding RNA. And then these batch-related oligo are going to allow you to do a specific pull down of this long non-coding RNA that interact with RBP, so RNA binding protein or DNA. And we either isolate the protein, the RNA, or the DNA in a genome-wide manner to actually find out where they interact in the genome. And this is something we've done now for several of these long non-coding RNA. It's still a work in progress, but we were happy to see that the methodology worked. We are able to pull down a DNA sequence when the long non-coding RNA is present. We cannot pull down any DNA sequence when the long non-coding RNA is not present at different stages of the life cycle. And this is an example for the TAR, a long non-coding RNA that supposedly interact with the telomere end, and this is chromosome 1, 2, 3, and you can see the telomere end are here in orange, and then this peak here really co-localized nicely with the telomere end. So we are happy to see that our methodology definitely works, and we find some nice anti-correlation between when the long non-coding RNA is present and when the gene is transcribed. So we think that actually really important to recruit the right chromatin modifying enzyme at the right moment. So again, this is a work in progress. We're doing that for a lot of different new long non-coding RNA and trying to find a functional role for all of these long non-coding RNA. So in conclusion, we have developed a sensitive approach to identify novel long non-coding RNA in Plasmodium. We also validate their subcellular localization at single cell level using RNA fish and CHOP-seq to specifically analyze long non-coding RNA DNA interaction. And these long non-coding RNA certainly have a role in chromatin structure and transcription regulation by encoding protein complex to promote region of target gene. So very nice correlation between all of these long non-coding RNA and repressive of several of these genes. So we're now doing CRISPR-Cas9 technology to actually really validate the function and the essentiality of this long non-coding RNA. But these long non-coding RNA usually don't work alone. They work in interaction with protein. So we also developed this genome-wide approach to identify all potential protein interacting with chromatin in all of these apicomplex parasites. So again, I'm not going to get too much into details, but I'm going to just tell you that we developed a novel pipeline to identify a lot of these chromatin-associated protein that we call CAD, chromatin-associated protein in apicomplex parasites. And all of these protein by doing domain search and HHM search within all of this genome, we identified a large amount of this protein that have, of course, a function into chromatin structure and gene regulation. The amount of CAD protein that we find in different genomes, Plasmodium falciparum, Vivax, Toxoplasma, Trypanosome, Leishmania, so we again analyze a lot of different genomes. And it's approximately similar number with the exception of Trypanosome and Leishmania, Trypanosome that don't have a clear transcriptional regulation in their genome. So I'm not going to get too much into this detail, but we're happy to identify a lot of this protein. When we actually cluster all of this domain, all of this protein together, we find these CADs that are rich in unicellular organisms, Plasmodium falciparum, Trypanosome, Leishmania, Saccharomyces cerevisiae, some that are specifically enriched in apicomplex and parasites, and some that of course are rich in humans or plants. So we have this very large enrichment of different type of CAD within different genomes, and of course we're interested in this family that can be targeted specifically for apicomplex and parasite compared to human or mammalian. But it's great to do in silico, but we really wanted to make sure that all of this in silico search was true. So we developed another a methodology to do chromatin enrichment for proteomic CHEP, where we cross-link all protein with the DNA and specifically purify this protein and do a different MS search. And again, we were extremely happy to see that this methodology worked. We have a very nice overlap between in silico versus our experimental approach, different type of CAD across the different life cycle stage. We also have a very nice correlation coefficient between biological replicates, and a lot of this protein we know are of course involved in chromatin structure and transcription regulation, such as histone, DNA binding, and so on. So this was great, and then we validated a couple of those at the functional level, where we designed specific antibody to validate that they're indeed interacting with the chromatin. So we took a SMC, which is a chromatin complex, and then we also were interested by plant-like protein from this cluster 1, and this chrome-like protein that seems to be really important in plant development to maintain the nucleus integrity and membrane of the nucleus in the plant. So we designed antibody. It's really hard to see, but we clearly show that these protein interact or at least co-localize within the nucleus at different stages of the cell cycle. And then we did a ChIP-seq chromatin IP to localize this protein in the genome, so SMC. And we were happy to see that this is actually nicely co-localization with the centromere. So very important for the centromeric function and cell division in the malaria parasites. The antibody didn't work, unfortunately, for the crown protein-like, but we're working on it and trying to generate a CRISPR-Cas9 system, a tag protein. So conclusion part 3, we generate comprehensive in silico overview of chromatin-associated protein in Plasmodium and other apicomplex and parasites. We identified parasite and plant-like specific component that we have validated. We've been trying to generate knockout without any success, which means that this protein may be essential for the malaria parasite, so we're trying to reproduce our results. But we think that they are extremely important to regulate chromatin structure or gene regulation, and work in progress to really validate this as a lamina-like protein in the malaria parasite or apicomplexan parasite. So where do we go from there? Now we have all of these long non-coding RNA candidates and these chromatin-associated protein candidates. We are using the CRISPR-Cas9 technology to specifically knock down or knock out features such as the super domain structure that we observe at the chromatin level, this specific long non-coding RNA that seems to be important for sexual differentiation, and this plant-like protein that's have a very important role in plants. We think that they could be really good drug target in apicomplexan parasites. So as I said, this is work in progress. Plasmodium falciparum is 80 rich, hard to get. So far we have been constricting all of the vector to do CRISPR-Cas9, and we've generated several transfections, but we're waiting for the parasite to come back. So here it is, so work in progress, still working on trying to understand and identify the major molecular component driving the parasite life cycle progression with the hope that we'll be able to stop the transmission of this disease from mosquitoes to human or human to mosquitoes. So I would like now to thank people that have been working on all of these projects, people in my lab, but I have also a lot of different collaborators around the world that send me different samples. Bill Noble has been my long-time computational biologist collaborator. Jean-Philippe Baer, which is a mathematician that have been helping us in the 3D model. I received samples from the recent lab for Toxo. Ferhat has also been helping a lot in computational data analysis from Malaria Institute for Immunology. I received samples from David Fiedler, Columbia University, John Hopkins. Most of the sporozoites come from John Hopkins and also Thailand. People have been helping me in terms of transfection and Plasmodium knowlesii come from Marek Galimski at Emory University. Babezian sample come from Shukri Ben Mamoun, and all of my funding, NIH, CDC, and USDA. Thank you.